Thanks, Greg. Thanks, everyone. This is kind of the lighthearted, fun talk here. So the initial title that I had for this was actually Who Needs Technicians? Robots Can Do It Better. And the USGS approving officials kind of took me aside, like, you know, this is kind of offensive. So anyway, this is where we landed. But this is a very cool project in which we got to pilot the use of some cool, I don't know, novel eDNA gadgets. So that's, you know, really what we're going to be talking about here. So, you know, this goes back about 2 years now. I heard from some folks in the National USGS ReadyNet project. They said, hey, you know, would you be interested in an opportunity to pilot a possible eDNA robot or autonomous sampler? And of course, I was all for that. And immediately I knew what I wanted to do with it. I wanted to put it in a USGS stream gauge where we have all the awesome air and discharge and turbidity and temperature data to help us understand the eDNA data we'd be getting. And the gauge I wanted to use was on the Hudson, just upstream of Green Island, what we were just talking about. However, this is what the gauge looked like in spring of 2023, and you know, the kids would think the word they would use is a little janky. It was looking right. So this was clearly not a suitable location, you know, to do the work that we had to do. So a big shout out to our stream gauging folks and Mr. Wells. If you could just give me that click, please.
Green Island gauge removal, part 2.
There's no way related to any data that you need to see, but it's just, why wouldn't you show this?
Nice.
All right. So, I think I need another click out of you there, Scott.
Not Green Island.
There we go. Oh, we're good, we're good.
Show it again.
There you go.
Green Island.
Oh, all right. We're a little touchy, but I think we're good. So a month later, we had a beautiful work environment, a cleaned up area, and a lovely stream gauge. Wow, very touchy here. I can just sit at the computer. Hang on. All right. So this was part of a national effort conducted 4 separate case studies really across the U.S. So the Hudson was the first one that was run, then Cherry Creek, this is a small trout stream in Montana, then Loggers Creek in Idaho, slightly larger creek, and then Big Piney River in Missouri, pretty big water. So all 4 of these tested the same eDNA autonomous sampler using different qPCR targets, you know, different species that were being assessed. These were generally 7-day deployments, with the robot being asked to take a sample every 3 days, with a few exceptions here. So, you know, the objectives here, I guess the first, you know, really twofold, right? The first question was, okay, you know, technicians are, you know, limited in terms of, you know, how many hours they can work at what times a day and things like that. The robot has none of those limitations. Does that even help us in any meaningful way? So, like, what are we missing at night, right? The robot's happy to take a sample at 2 a.m. if you wanted to. How severe is the temporal variability? So if we're just cranking out samples one after another, are we seeing large changes in DNA concentrations for our target species? If so, then that means taking a single sample at a point in time may not be representative really of the bigger picture. And of course, what's the effect of our environmental covariates, right? If I sample after a big rainstorm or came up a foot, you know, what does that mean as far as detection probability or eDNA concentration? And probably even more relevant to folks who use eDNA in their job today is, you know, benchmark it against the established methods, whether it's your Smith-Root backpack or your hand pump or Citizen sampler, you know, whatever you're using. How well does the robot do in terms of detection rates and in terms of DNA concentrations? So some specs on the robot. This is a Smith-Root prototype device that we had the opportunity to test. Everyone will sigh in disappointment when I tell you it is archival, so this is not analyzing samples. This filters the sample, it preserves the sample because it's using the self-desiccating filters, and it can sit indefinitely until a technician arrives, but this is not extracting or amplifying DNA. Very quickly, we programmed it to do 5 liters per 5-liter flush and then a 2-liter sample. Again, 8 samples per day. The robot does have fixed permanent water lines, both internally and externally, running to the river, and that may become relevant later when we start to talk about contamination. Field blanks, kind of difficult. You actually have to remove the water intake from the river, immerse it in a bucket of DI water, and then flush DI and take your sample. This is going to be a struggle the whole way here. All right, so this is the Hudson River. This is the first set of data, and there's more data here than you could ever possibly hope to glean in a couple of minutes, so I'm just going to very quickly say these black dots, that is the average DNA concentration from a given sample. It's the average of the 5 PCR reps, and then the error bars show you the range of the PCR reps. On the top panel, you have discharge from the stream gauge. Bottom panel, you have turbidity. So I guess the two take-homes here. First, these eDNA points, how much variability do we see from one 3-hour sample to the next? And the answer is a lot. This is log-scale y-axis. We see, in some cases, like an order of magnitude difference in DNA concentration from one sample to the next. So that's frustrating. What do we see in relation to the environmental covariates? We actually see as discharge and turbidity increased after a July 4th rain event, we actually see higher DNA concentrations after that period associated with increasing flow and discharge. That is counterintuitive and not what is supported in the literature, where you would expect a dilution effect instead. This was not a surprise. So the y-axis is showing you the proportion of our target volume that we got. So we were trying to get 2 liters in every sample, and when turbidity was low, meaning less than 5 FNUs, we were able to get our target volume almost every single time. As turbidity increased to 10 or higher, we were generally getting less than 50% of the target volume. So this basically means the robot's not immune to filter clogging, right? Just like any other filtration method. So not a big surprise. On the benchmarking, sorry, I can talk through it without a slide, that doesn't bother me. For the benchmarking, wow.
Want me to do it from here?
You're going to be doing a lot of clicking. For the benchmarking, oh, one more click there, Scott. Yeah, I guess you'll be the clicker, sorry. One more. We compared 3 different methods. So it's the auto samplers, the black dots, the Smith-Through backpack sampler is the pink dots, and the blue dots, that's a hand pump using a glass fiber filter. So the backpack and the auto sampler, both using 5-micron PES filters, they're identical. And essentially what we see here is the auto sampler consistently picks up higher concentrations of DNA than the backpack sampler. The hand pump not only is getting low concentrations, this is again using a glass fiber filter, but it is also even struggling to get detections in some instances. Another click, please, Scott. All right, so switching to our small trout stream, high elevation in Montana. Our target qPCR critter here was a West Slope cutthroat trout, which hopefully is what that is. And this was a 7-day exposure similar to what we had done before on the Hudson. We see eDNA concentrations a little bit more stable. We don't see them bouncing around quite as much from sample to sample. They had a small rain event, I believe around the 19th or 20th of August, and DNA concentrations responded in a more predictable fashion in which they became quite variable, but they also went down generally. The benchmarking effort, they took a pair of manual samples every day when they tended the robot. The detection rates remained 100% in this system, but the DNA concentrations were considerably lower in the manual collected samples. Go ahead, Scott. Loggers Creek in Idaho, we were looking for rainbow trout on the top, and we were looking for a native unionited mussel western pearl shell on the bottom. This data set is a little bit more, mundane in that we don't see major differences between the manually collected samples and the robotic samples. Go ahead, Scott. Finally, Big Piney River. This was part of a larger experiment that Katie Klimas's team led. The vertical line here is showing you the addition of a frozen lake trout slurry into the river. Lake trout are not present in the watershed, so this represents a novel DNA source. As this block melts, it releases novel DNA that otherwise wouldn't be in the system. So the autonomous sampler was programmed to just sample like crazy before and after, and they took a lot of paired manual samples. Those were grab samples for centrifugation because that's their prevailing method in that neck of the woods. And we see exactly what we would expect, right? Negatives before the slurry addition, then a couple low positives in the very beginning, and then about an hour later, DNA concentration goes through the roof with both methods and gradually tails off. The only difference here is that the centrifuge samples, because they're small volumes, we see a lot more variability between samples. Go ahead, Scott. So findings and conclusions. So, you know, we use this autosampler in 4 different study areas across the U.S., ranging from a large river at essentially sea level to a high elevation trout stream, and we programmed it to do particular things, and then we analyzed those samples for target DNA sequences. And in all instances, you know, it worked at the most cursory level. Not to say it wasn't without challenges, but it collected the samples when we told it to, and those samples retained DNA that we could detect. It took about 10 to 15 minutes, closer to 15, to collect a sample. So it's about 6 minutes to do the flush, in which we're blasting river water through the system to try to flush out any residual water or DNA that might be in there. And the remaining 9 or so minutes was to actually collect the 2-liter sample. We know that there was no specialized training needed because Austin Thomas was supposed to fly out from Smith Root to help us, and he missed his flight, and we did it all over the phone. So anyway, the benchmarking, comparing the autosampler to established methods, right? Each study area had their own established methods where essentially, you know, their prior monitoring had been using a particular method and filter type. Benchmarking showed that detection rates were quite similar between the manual methods and the autonomous samples. However, DNA concentrations for at least 2 of the studies were actually notably higher in the autonomous samples. I was very excited for day versus night. I thought this might be, you know, in the absence of UV radiation right at night, maybe DNA concentrations could be higher. We found no evidence for that whatsoever. Day versus night was not really an exciting comparison. Between sample variability was very high, and this kind of stinks, honestly. So, you know, a single sample taken at 2 p.m. might have a very different concentration of DNA from a sample that was taken 3 hours later, potentially. And this is problematic because if you're taking a sample once a month as part of some long-term, you know, monitoring plan, how representative is your sample? For a detection rate, maybe it's not bad. For a DNA concentration, you probably need some replication to really get a handle on what it means. Discharge. This was an interesting covariate for us because in the small trout stream, when discharge went up, we believe we had a dilution effect and we saw lower DNA concentrations. On the Hudson, when discharge went up, turbidity went up too, and we actually saw higher DNA concentrations. We don't really know why, but we believe it may have to do with resuspension of sediments that have DNA bound to it. We certainly haven't tested that hypothesis, but if anyone has a better idea, we're all ears. Turbidity decreased the amount of water we could get to pass through a filter. That's not unique to the robot, that's just a function of, you know, filtering in natural waters. All right, this is probably the biggest point I need to make before we wrap up here. One more quick, please, Scott. So, if you look closely at those graphs, you might have noticed that there were field blanks embedded into some of them. This is a snippet I took out of our manuscript because I wanted to make sure I got this right. 4 of the 5 field blanks that were taken in the midst of an experiment. We're not talking about blanks that were taken, you know, in the setup phase. This is after we're already filtering and sampling, and then we take a break to do a field blank. 4 out of those 5 tested positive for the target DNA sequence. These are field blanks, right? This is deionized water. So we've already done a 5-liter flush of the system with DI water, and then we filter 2 liters of DI, and we still got detections in 4 out of 5. Now, the concentrations of DNA were 1 or 2 orders of magnitude below what we were getting in the preceding and proceeding samples. So it's low-grade contamination, but it suggests that those permanent water lines probably are retaining enough DNA to be a concern. Now, I do want to mention that if you're using this technology to screen for a species that's presumed absent, like you're looking for snakehead on the Hudson or something like that, this probably doesn't matter because there's no contaminating source. But if you're looking at changes in DNA concentration to infer when striped bass are reaching their peak spawning period in the Hudson, you know, then this is potentially a confounding factor. Go ahead, Scott. So I'm pleased to say, we were able to get this combined effort published in Environmental Science and Technology a couple months ago. So it's a Herculean effort to get over the finish line, but very pleased about that. Go ahead, Scott. And all the underlying data from each case study are published in a USGS data release as well. So I want to thank the USGS ReadyNet project. It's part of the Ecosystems Mission Area. They've been an awesome partner to work with on this. Smith-Root's been great. They let us break their stuff and answer the phone at all hours of the day, and they've been really positive to work with. Numerous students and interns, some of those folks who are here today who have helped man the robot, and also just want to thank the Water Science Center for helping us with all the stream gauging stuff on really short notice with unreasonable requests. So go ahead, Scott. I think we're home free here. But yeah, happy to field any questions. Thank you.